So, welcome you all once again for the 22nd class. So, the title of this class is monoclonal antibody production. Okay. So, in the previous class, we decided that we will be exploring all the different recognition elements like monoclonal antibodies, single chain antibodies, aptamers, peptides, fast display. So, towards that we talked about the monoclonal antibodies as of now. So, today we will talk about how these monoclonal antibodies are being produced. A brief understanding of monoclonal antibody production is essential for the next generation of biosensing experts, because I believe that it will bolster their capabilities and will help them to understand and use this technology with far more perfection. Now, in terms of monoclonal antibody production, the very first step is out here: immunization of mice and isolation of splenocytes, which is you are immunizing the mice with an antigen. So, you take a foreign material, you are injecting it in the mice as the drawing says, and later their blood is screened for antibody production. The antibody producing splenocytes are then isolated for in vitro hybridoma production. So, whenever you are injecting an antibody or an antigen or a foreign element, when you are injecting a foreign element which is called as antigen into a host, this host could be anything. It could be a mice, it could be even human. The body will be producing antibodies against this antigen. So, this is exactly what we are talking about, and splenocytes are the ones which are generating the antibodies. So, preparation of myeloma cells, this is stage 2. Myeloma cells are immortalized cells. Myeloma are basically something like a cancerous cell. Once fused with spleen cells are the splenocytes. If you come across a word splenocytes, means spleen cells. Cytes means cells, spleno means spleen. Okay. Splenocytes. So, now these primary splenocytes or regular splenocytes of your body have a limited life cycle. Now, in order to have a unlimited production of antibody, so say for example, I inject an antigen into a mice or a human, then I isolate the antibody producing splenocytes. So, I know this particular splenocyte or spleen cell is producing the antibody. Now, what I do after screening that particular cell, isolating that cell, I will make a clone of it. I will immortalize that cell. How I will immortalize that cell? Using cell technologies, I will cross it with a cancerous cell, which is a myeloma cell. By fusing it with a myeloma cell, what happens is that this particular splenocyte producing that specific antibody is now immortalized. I can freeze it, I can thaw it, I can grow it, I can keep it for years together. After 30 years, 100 years even, I can pull it out and make it work. And I will be knowing with absolute certainty if the DNA is intact, if there is no freezing damage of DNA or something, then that particular splenocyte will produce that specific monoclonal antibody. Okay. So, now coming to the second step again, preparation of myeloma cells. Myeloma cells are immortalized cells that once fused with the spleen cell can result in a hybridoma capable of unlimited growth. Myeloma cells are prepared for fusion. So, when we talk about the hybridoma technology, it is basically a hybrid between myeloma with a spleen cells or antibody producing cells. Okay. And as I mentioned, always remember it like this. Sites stand for cells, and here it could be anything. It could be some other tissue. It will be site. Okay. Now comes the fusion. The myeloma cell are and isolated splenocytes, as I mentioned, are fused together to form hybridomas in the presence of polyethylene glycol, which causes cell membrane to fuse. So, you are fusing both of them. So, you get something like this. Now, clone screening and picking. Clones are screened and selected on the basis of antigen specificity and immunoglobulin class. Antigen specificity means which particular antigen they are neutralizing. So, you know that this particular clone is working in that direction. Now comes the functional characterization, where you confirm, validate and characterize, that is ELISA, each potentially high producing colonies. So, there are multiple colonies which are produced. So, you have to check for the fidelity in terms of confirmation, validation and characterization. Scale up and wean, scale up clone producing desired antibodies and wean off selection agent. Okay. You are slowly narrowing down and you are scaling it up and weaning out the unnecessary ones. Finally, the expansion. Expand clones producing desired antibodies in the bioreactors or large flask. So, this whole monoclonal antibody production process is a cellular engineering marvel, where using different cellular technologies, you are fusing cells, you are producing them in large number, you are isolating them, you are propagating them. It's an industry of its own, and these industries are now merging with the biosensing industry, and a new change or a wave is coming in the whole area of bioengineering, biodesign, where lot of these kind of sensors are being made because you have easy access to a lot of antibodies which are extremely specific for a specific pathogen. Now, in terms of the infographic illustrating the process, here you have the antigen. In order to give you a better idea of those people who have come from different background or who never got a chance really to visualize the whole thing, immunization of mice with a specific antigen to stimulate antibody production, isolation of antibody screening plasma cells. These plasma cells are then fused with the myeloma cells and fusion and generation of hybridoma. So, now at this stage you have to screen which one has all the potential of what you are looking forward to. Then comes selection in HAT medium. So, do not worry about HAT medium or ELISA screening, because these are cellular technologies where you use a specific medium to grow or select a specific clone or population, and there are other enzyme linked immunoabsorbent assays for screening, and thereafter expansion of the selected hybridoma to produce monoclonal antibodies. Okay. So, this is one simple way of it. There are other ways which will be coming, but this is an overall understanding you will be needing about monoclonal antibody production system. Now, talking about the steps: immunization of mice and isolation of splenocytes. Mice are immunized with an antigen and later their blood is screened for antibody production. The antibody producing splenocytes are then isolated for in vitro hybridoma production. In vitro means you are producing it outside the body. So, first part was inside the body, part 2 is outside the body. That is why it is called in vitro. Preparation of myeloma cells. Myeloma cells are immortalized cells that once fused with the splint cells can result in hybridoma capable of unlimited growth. Myeloma cells are prepared for fusion. Next comes the step of fusion. Myeloma cells are isolated splenocytes, are fused together to form hybridomas in the presence of polyethylene glycol, which causes cell membrane to fuse. Then comes clone screening and picking. Clones are screened and selected on the basis of antigen specificity and immunoglobulin class. Then comes the functional characterization, where we do the confirmation, validation, and characterizing them using ELISA or potentially high producing colonies. Scale up and wean, as we mentioned, scale up the clones producing desired antibodies and wean off the selection agents. And expansion, expand the clones producing desired antibodies, bioreactor or large flask. So, this is the overall scheme of things or flow diagram which one has to keep in mind while producing monoclonal antibodies. So, if you summarize the whole thing, the process of immunization, preparation of myeloma cells, fusion of the myeloma, clone screening. Now, for clone screening, there are a lot of technologies which have currently being used. There are different vendors like mammalian colony pickers, then you have clone select imager, single cell dispensers, then you have mammalian colony picker and monoclonal assurance with the Z stack. There are a series of, as I mentioned, there are multimodalities now which are being used for clone selection. Otherwise, it used to be a very tedious process. Well, you have to really go under the microscope and observe each and every colonies and you have to pick it up. But now this whole process has been automated. But the basics are the basics. Basics hasn't changed. It is just for ease and time saving. The technology has been automated over a period of time. From here, you have the functional characterization. Again, a huge amount of automation which has happened. Like these were not there, at least I remember during my postgraduate days when these technologies were picking up back in 90s. All these automation and use of robots and all these things were just brewing in the R&D wings. But today, almost now 30 years down the line, things have changed. Things are much more automated, much more simple, yet the basics are the same. So, always remember the basic, try to understand the basics. If you do not have these automated systems, how you can operate under this situation. And then comes the scaling up. Again, there are a lot of very good quality bioreactors which have developed, and the expansion using different kind of multimodal equipments. So, overall if you look at it, this is very basic fundamental process. You inject an antigen, the body generates its antibodies. So, you isolate the antibody producing splint cells, you take these splint cells, isolate the specific antibody producing ones which you are looking for, because different splint cells will be producing different kind of antibodies. Now, that particular splint cell, you fuse it with a myeloma cell using polyethylene glycol, where the membrane merges, and this we call as a hybridoma. This hybridoma is being screened through for production of monoclonal antibodies. This is the whole spectrum of things. Now, during this process, there are zones where there is lot of room for automation in terms of how you are selecting the clones, how you are screening the clones, how you are propagating the clones, how you are weaning off the unnecessary, or at that particular instance, the one which are not important ones, and how you scale up or jack up the production of these clones. So, these technologies have made monoclonal antibody production a much more easier technique. So, this particular technology is hybridoma based technology. There are other technologies of monoclonal antibody production. What we will be discussing is called phage display and further selection methods, which are a little bit more molecular biology intensive techniques, unlike hybridoma, which is a cell based techniques which are being used. And further, this monoclonal antibody production has revolutionized the whole field of immunocytochemistry, especially in developmental biology and all other field of disease progression, because whenever we see a signature, so these are all recognition elements. Whenever we see a specific signature, we produce that monoclonal antibody. So, in a way, immunology has equipped us with very specific tools of identifying pathogen, identifying how the pathogen react and what kind of reactions it brings about, and how the anti-molecule towards the pathogen or an antibody fights against it, and where the antigen changes its properties and evade or trick the antibody system. And we have learned a lot over last 3 or 4 decades in terms of AIDS virus, in terms of prion disease, in terms of COVID, in terms of some tricky cancers, in terms of neurodegenerative disorders. All these situations are situations where the body fights. And whenever we say body fights, means body is producing antibodies. So, it is always an antibody-antigen reactions which brings about the orchestra of body fighting for its own survival. And this technology is an asset for biosensor production. So, in overall, the basics remains the same. If we go back and check this, this is the step 1 where the body is doing its own work of fighting against the particular antigen which is getting injected, isolation of the plasma cells, crossing of the plasma cells with myeloma cells, fusion, then selection, and then expansion, and further producing these monoclones, right. So, this is the overall spectrum of hybridoma technology for monoclonal antibody production. And if we look at in graphically, this is the fusion process, clone screening process, cell growth, antibody interaction using affinity, specificity, and functional screening. And these are the different technological breakthroughs in terms of mammalian colony picker, automated cell imager, high content imager, and microplate reader, imaging cytometer, and a high content imager. So, if you look at it, this bottom line out here are scaling up technologies, which is revolutionizing the hybridoma production and saving time and energy. Okay. So, in the next class, we will move with the next technology for monoclonal antibody production, which is a phage display technology. Thank you.